It's 2 o'clock, and welcome to everybody. My name is Victoria Harden, and I'm the director of the Office of NIH History, and we welcome you to the Briggs Seminar, and in particular to this Stetton Fellow lecture by Dr. Leo Slater. I wanted to make a few remarks as we get started about the Stetton Fellowship itself, which was created by the Advisory Committee to the Office of NIH History as a living memorial to Dr. DeWitt Stetton, Jr., who founded our museum and who spent most of his career here at the NIH as a scientific director of one institute, the director of another institute, and he had a different title, but he was essentially the Mike Gottesman of his time, the deputy director for intramural research in Building 1. Dr. Stetton died in 1990, and the first Stetton Fellow came in 93. And we've had quite a number in partnership with individual institutes whose work they have looked at. This year, Dr. Slater's work has been on the National Institute of Allergy and Infectious Diseases and its work in malaria, and I want to thank the NIAID and especially Dr. Catherine Zune, who is with us today, for supporting Dr. Slater's work. Dr. Zune is the acting director of the Division of Intramural Research at NIAID, and she's the lady standing because she's got a foot problem. But I've asked her to come make some remarks, so we'll let her do that for the moment.
Okay, well, thank you very, very much. It's a pleasure to be here today. And over the years, I've had a number of opportunities to interact with the History Office and particularly with Victoria. And it's been on a number of levels for a number of occasions. And I think the opportunity to document our history is extremely important for the NIH, and particularly in today, I'd like to focus on NIAID's impact. As you know, the history of any given scientific program is really important that we document it and have a written record for the public in order for them to appreciate the investment that they make in the NIH research programs, in this case the NIAID research program, to help further the public health. And this is really something that I think I really am very, very supportive of, and I know NIAID has been interested in this for quite a long time and has its own really rich history of recording its history and accomplishments. Dr. Harden, as the NIH historian, has worked on a wonderful document outlining the accomplishments and history of the Rocky Mountain Laboratories. This is, for many of you who may not know, is a laboratory that's out in Montana, in Hamilton, Montana, in the Bitterroot Valley, and a beautiful place if you've not been there, and really the home of Ricketts, who discovered rickettsia out there. And so a very rich history now, really a major source of intellect in terms of infectious diseases, both bacterial and viral infectious diseases. We also, under the auspices of Dr. Harden, have a history of HIV and AIDS research that she has done, which I think over time, I think is actually going to be a wonderful historical record of this serious public health challenge and its role with NIH, in particular NIAID. And finally, she has co-edited a book on the centennial of the intramural research program of NIAID, dating back from 1887 to 1987. And I dare say we will be working on biodefense and emerging infectious diseases shortly, so we get the history right. But today, our focus is going to be on malaria. And I just wanted to take a few comments to say how much we support and really believe that the Stetton Fellowship Program is such a wonderful idea. And we really like to see bringing together the historians with the working scientists so that we can meld and bring to the public the understanding of how the history of a particular disease or a particular agent or area of research melds with the scientific productivity and really leads to outcomes. And I think today, when we talk about malaria, it is really an excellent case study of this. And I'm just totally pleased that Dr. Leo Slater's research is on the history of malaria, and really to highlight both the predecessors of this research as well as the current work going on in our laboratories. And I'd like to give an NIAID intramural program particular emphasis both to the Laboratory of Malaria and Vector-Borne Research, as well as our Malaria Vaccine Research Development branch. And I think both of these are really good examples, and I'm looking forward to the work that we'll hear today from Leo. Now, this all stems from actually some very important work done by James Shannon. And many of you know Dr. Shannon. Building 1 is named after him, so he is really one of the giants in the NIH community. And some of his first work actually was on malaria during World War II. And I think this kind of history integrated with the science today will actually really be very enlightening, and I'm really thrilled that Leo is going to give us this perspective. As you all know, malaria today is still a major global contributor to mortality and morbidity around the world, and it's our scientists that are really looking to alleviate this disease and trying to find new vaccines and new medicines to really take this to a new level that can really give a better quality of life to individuals around the globe. And I'm hoping that with the additional information from both the historical and our working knowledge today, as well as our future research in malaria, we may really find things to really give relief to this devastating disease. So with that, I'm going to turn it over to Dr. Harden. Thank Leo, thank Victoria very much, and it's an honor to be here to hear you present the Stetton Lecture. Thank you very much.
Well, as Dr. Zune said, and anybody who knows me knows, infectious diseases is my particular interest in research. And curiously enough, Leo is the first Stetton Fellow to work within NIAID, so I was thrilled. And then the next year we got a second one, Dr. Lisa Walker, who will be giving us a talk next year. I'm just very thrilled that the oldest institute on campus is now getting some additional historical treatment. I also want to put in a plug before we get to the lecture for a conference that we're holding on December 5th and 6th, at which Dr. Slater will be speaking, by the way, on malaria. It's a 2-day conference called Biomedicine in the 20th Century, Practices, Policies, and Politics. And we're bringing together some of the senior historians across the country to address the rise of biomedical research in the 20th century. It'll be in Lister Hill Auditorium. It's free, of course, but you do need to register because we have a limited number of seats. It's on our website on the home page. If you go to history.nih.gov and click on the conference, you can sign up. I hope more of you might be able to come to at least part of it. Now, turning to today's talk and today's speaker, Leo Slater, like many other Stetton Fellows, comes to NIH with backgrounds in both science and history. He took both his BS degree at Brandeis and his MS degree at Stanford in chemistry. He then worked for 4 years as a chemist responsible for the synthesis, elaboration, and purification of peptides at Schering-Plough Chemical Research Division in New Jersey. In 1992, however, He decided to move from researching in the laboratory to studying the history of research in the library. He enrolled at Princeton and took both MA and PhD degrees in history with a dissertation on organic synthesis and R.B. Woodward. And then from 1997 to 2001, Dr. Slater worked as a historian and administrator at the Chemical Heritage Foundation in Philadelphia. In that year, he began to focus on the history of malaria research in the Malaria Research Institute at the Johns Hopkins University, with joint appointments in the School of Public Health and the Department of the History of Science, Medicine, and Technology. He continued his research and writing during a year at the Max Planck Institute in Berlin and has now completed a book manuscript on malaria and war, which examines the U.S. malaria research effort during World War II and analyzes scientific thinking about what constituted an adequate research model for disease. During his year at the Office of NIH History, he's been able to expand his knowledge of malaria research to include NIAID's contribution. And his talk for today will assess that work. Dr. Slater.
No? How about now? Well, that's good. Thank you. Thank you, Dr. Zoom. Thank you, Dr. Harden. I'm going to tell two short histories today of two projects. Each lasted several years. The two projects were separated by about 20 years. The first took place in the early 1940s during World War II, the second in the early 1960s. These are, as has been suggested already, part of a larger project I'm working on the history of malaria research at NIAID and its predecessor organizations. But what I think these two projects have in common more than just being about malaria research is that they're part of a longer tradition of service at NIAID and at NIH. The ability of the institution to react to public health emergencies in times of crisis and address those emergencies. So if we think in more recent decades as part of this tradition, in the 1980s and 90s, we had AIDS, HIV. In more recent years, the SARS epidemic, Katrina in recent months, and just in the last few weeks, a whole set of new initiatives regarding pandemic flu. So it's in this tradition that I want to try to look at these earlier episodes. Before I move on to the history, for those of you who are not malariologists, just a quick introduction to what malaria is. It's a mosquito-borne parasite. It's a more complex organism than a bacterium. It requires both an insect host, that is in the case of human beings, an Anopheles mosquito, and a vertebrate host, a human being in this case. There are 4 recognized species of human malaria, Plasmodium falciparum being the most deadly of them, and Plasmodium vivax will be the one I'll be focused on today, and then 2 others, Malariae and Ovale. What I really just want you to take away from this diagram today is 2 things. That the parasite has a complex life cycle that involves 2 hosts, the insect host and the vertebrate host, and to procreate it needs to go through both those hosts. And secondly, just looking inside the human body on the right-hand side here, to recall that there is a liver stage to this parasite. I'll be talking a little bit about that. At the time of our first episode in the early 1940s, this liver stage was not known, but there was known to be a latency period in all these. And all terrestrial vertebrates have associated malarial parasites, all orders do. So it's a fairly common disease across the animal kingdom. The 2 episodes I'm going to be talking about are going to be focused to the first and the last locations on this list. This is a partial list of field stations and remote laboratories of NIAID and its predecessors where malaria research was conducted. These you can pair up in a number of ways. Columbia, South Carolina, and Milledgeville, Georgia, were both laboratories associated with state mental hospitals where the neurosyphilis patients were treated with malaria therapy, and I'll be talking a little bit about that in a minute. Seagoville, Texas, and Atlanta, Georgia were the site of federal penitentiaries with prisoner volunteer projects where research was done there. And the 2 outliers are Memphis, Tennessee, which, as the people who worked there described it, was the pure research laboratory. That is, there was no human malaria at that research facility. they primarily studied the biology of the parasite using model systems such as monkey malaria. The final one on the list, Kuala Lumpur, obviously stands out as being the most remote from where we are now in Bethesda, and one of the later ones also. As I said, we're going to focus primarily on the first and the last of these. So let me start with the Columbia Lab. The Columbia Lab was set up in 1931 by Louis Williams and Bruce Main, two eminent malariologists from the first half of the 20th century. Williams directed virtually all of NIH's malaria research during the 1930s. In 1937, Martin Young joined the laboratory. And when Dr. Main passed away in 1941, Martin Young became the director of the lab. Two other names I've included on this chronology just to introduce them as characters because they'll come up later are G. Robert Coatney, who joined the staff in 1938. He spent a year or two in Columbia before coming here to Bethesda to direct the malaria chemotherapy research during World War II. And at the bottom we have Don Iles did a brief stint here in Columbia. This was not his first NIH posting nor his last. He'll come up again in reference to our second episode. This is a picture from a slightly later period. It gives you a picture of the staff. There's really just two people I want to point out off the South Carolina staff here. One is obviously Martin Young, who was director, seated at the right. And although she's not part of our story today, I think she's part of our theme today. The woman sitting in the center in the dark dress with the three light stripes is Amy Wilcox, and she literally wrote the book on the microscopic diagnosis of malaria. It's NIH publication bulletin number 180. It went through a number of editions. And during World War II, she trained hundreds of people to diagnose malaria using the microscope. They in turn trained others. And I think it's a nice example of how someone stepped up in a time of emergency to deal with this public health threat. These are the grounds of the South Carolina State Hospital. As I said, it was chosen because of a cooperative venture there involving malaria therapy for neurosyphilis. Neurosyphilis, that is syphilis of the brain and spinal fluid, was an extremely debilitating and terminal disease, extremely terminal. It was devastating, and in this period, in the 1930s, 40s, the first half of the 20th century, large percentage of these state mental hospitals around the world, large percentage of their patients were neurosyphilitics. In the 1920s, a new therapy was developed, malaria therapy, in which these patients were actually deliberately given malaria either by blood transfusion or mosquito transmission, and the disease was allowed to run its course for days or weeks, or for its natural course for a long time, that is. And it was believed that the repeated peaks of fever caused by malaria provided a therapeutic effect against the syphilis. Depending on what numbers you look at, something on the order of a third of patients were actually improved by this therapy. Another third of cases showed an arrest of the disease, and another third showed no therapeutic effect. But it was one of the few and best therapies available. Now, it should be noted that for most of this period, the adding on of a research agenda to this therapy was considered a normal part of the therapy and not a separate issue. What I want to talk about specifically at Columbia is the Imported Malaria Studies program. I should say that this is just one part of a larger set of initiatives begun as soon as the U.S. entered World War II to address the threat of tropical diseases coming back into the country with returning servicemen. This project itself didn't begin until the fall of 1943. It was set up by the Army, the Navy, and the Public Health Service. Of course, the Public Health Service had as one of its primary missions to protect the country's borders from diseases coming in. And it was managed by Martin Young at the Columbia Laboratory. It was a joint project between the NIH and the Office of Malaria Control in War Areas. These institutions had a lot of flow-through of people and mission, so it was natural that they would be partnered in this. Two things I guess I should add about the Office of Malaria Control in War Areas. First is that, as its name suggests, its primary purpose was to protect military bases and training camps from malaria, because when the war opened, you had a lot of people, recruits, moving to these places, and you didn't want to debilitate your recruits as you were processing them with malaria. So they were there to keep these bases and the recruits free of malaria. And the second thing to note about the office was that it's the predecessor for CDC. As soon as the war was over, this office was converted into the CDC. And again, at that time, there was a lot of flow between the NIH and this institution. I'm quoting from one of Martin Young's papers here. The purposes of the program were threefold, primarily to determine if the mosquitoes that are native to the United States could transmit these foreign malarias that were coming back, or if they were bad vectors for this, then you'd have much less of a public health threat because the servicemen would be a dead end for the disease. If the local vectors could transmit the disease, then you have a more serious public health issue. As I said, I'm primarily talking about Vivax malaria here, but it's a fairly complicated species. There are many strains, what we might call genetic isolates today, and from different regions you had different virulence, different behavior of the parasites, and so the secondary issue was to try to sort out some of this biology of these strains, and finally to take whatever the findings were and figure out what impact that should have on the public health interventions. So to do this project, you needed four things. You need your local mosquitoes, you need facilities to raise these mosquitoes, you need research subjects, and you need, of course, the foreign malarias you're studying. Let me start with the mosquitoes. This is not a complete list of the species that were examined. These are some of the major vector mosquitoes that were looked at. The top two on this list, I put asterisks next to. These are the primary vectors for endemic malaria in the United States, in the West and in the South. Once you've captured these mosquitoes, you need to make more mosquitoes, which they do on their own if you give them the right environment. So you'd bring a mosquito in, you'd give your female Anopheles mosquito a blood meal and a pan to lay her eggs in, and then once you had these eggs, you'd move them to an area like this where you have these open pans of water where you feed and raise the larvae. The larvae are aquatic. You can see there's a pupa picker here, but we'll see in the next slide. Picking the pupa by hand. I have to get a drink of water for that. Picking the pupa. But just to note, mosquitoes are insects. They undergo complete metamorphosis, so that's a four-stage process: egg, larva, pupa, adult. And obviously, you can't contain the winged adults in an open pan like this, so they're transferred to these emergence cages, and you can just see on the right in that jar the freshly emerged adults. Once you have your next generation of your local mosquitoes, you want to see if they are able to transmit these foreign malarias. You transfer them to a feeding jar. When you have them in a feeding jar, you're going to need research subjects. So the research subjects are the soldiers, sailors, and Marines from whom these parasites were originally collected, generally once they came stateside and they were ill, the prisoner volunteers in Texas and in Atlanta, and the neurosyphilis patients at a range of hospitals who were undergoing malaria therapy. This is a typical feeding. So you have your mosquitoes. These are your fresh, new, clean, unfed mosquitoes. You feed them on a patient who has malaria, and you see if these mosquitoes become infected. Here's a close-up. You can just see the mosquito in the middle there feeding through the gauze at the top of the feeding jar. In association with all this, of course, you have a lot of microscopic work to do to diagnose and monitor the infections in your human patients, and also to dissect and determine the infection rates in your mosquitoes. These are two of the techs from the South Carolina lab, Marianne King and Elizabeth Horn, posing for this picture, working in the microscopes. And as I say, obviously you need the foreign malarias. The program did look briefly at Plasmodium falciparum. It looked at only 8 cases, and as you can see from this slide, I described falciparum as non-relapsing. What that means is, for Vivax malaria, when the parasites enter the bloodstream from the infected mosquito, you get a mosquito bite, they make their way to the liver. For falciparum malaria, all of these parasites in the liver are going to emerge into your blood and give you this disease. In Vivax, some of the parasites remain in a hidden form in the liver. As I say, the location of this hidden form was not known, but the fact that it was there was known at the time of this research. Because of that, it meant Vivax was much more of a public health threat. So if you have a Marine who gets sick in the Pacific Theater, he can be treated, he can be asymptomatic, his peripheral blood can be free of parasites, he can come back home and weeks or months later have a relapse and become a reservoir and a potential public health threat. So Vivax was a much more serious threat from a public health perspective. And so they collected strains from various theaters, from India, Burma, from the South Pacific, from the Mediterranean. And primarily they used as a control a local strain, the St. Elizabeth strain, which was first isolated and characterized right here in Washington, D.C. at St. Elizabeth's Hospital. At the end of this project, they made a very nice 18-minute color 16mm film. I'm not going to show you the whole film. I'm going to show you something from the beginning and something from the end. And this, in 2 minutes, is going to take you through cinematically basically what I showed you with these stills with better music and narration.
The female Anopheles quadrimaculatus is a recognized malaria vector in the South. Various types of malaria occur in this region. How well does this mosquito transmit these different malarias? How well will it transmit foreign malarias introduced by infected persons? How will our different malarias infect other species of mosquitoes, whether native or introduced? To answer these and related questions, a method of laboratory testing has been developed. The answers to these questions are essential for appraising the danger of importing malaria carriers and for applying proper control measures if they are necessary. The United States Public Health Service established a program to determine whether native anophelines can be infected by and transmit the foreign parasites brought into this country. These studies must be conducted with humans, since the malarial parasites which affect man do not live in lower animals. Parasites which will infect mosquitoes are obtained from patients who have primary or chronic infections of malaria acquired in foreign countries. Adequate supplies of the Anopheles species to be tested are reared on a large scale in carefully managed insectaries. Susceptibility of these mosquitoes to foreign malarias is determined by dissecting the insects at intervals after they have fed upon a person infected with one of the imported malaria parasites. Testing the ability of the species to transmit the infection to susceptible humans is more difficult, and few individuals are willing to expose themselves to experimental malaria. However, malarial infections have recognized therapeutic value in treatment of cerebrospinal syphilis. Mosquitoes infected with malaria are allowed to feed on such patients.
And they go on to describe here the cycle of transmission. I just wanted a quick slide in between here to give you an idea of the scale of this project. It lasted about 3.5 years. In the various locations each year, they raised about 1 million mosquitoes, dissecting tens of thousands of these. They collected U.S. mosquitoes from a range of places and raised them up in the insectaries to determine their viability as vectors. It was a collaborative project that involved 10 military hospitals and 8 hospitals that were either state veterans or private hospitals where there were neurosyphilis patients. And there were more than 10 papers published. Martin Young's group alone published 10 papers out of this project. Now I want to show you the second film clip from the end of the movie, taking you through the 6 steps used to determine transmission.
We have seen the 6 important steps in testing for transmission of malarias. 1, blood from the patient with foreign malaria is checked for presence of gametocytes to be sure that forms of the parasite which can infect mosquitoes are present. 2, anopheline females are allowed to feed on a malaria-infected person. 3, these mosquitoes are cared for during the 10 to 14 days while the development of parasites takes place, and are checked periodically to determine rate of development. 4. The infected mosquitoes are fed on non-infected persons to determine if transmission occurs. 5. The mosquitoes are killed, dissected, and checked to determine the percentage of infection. 6. The patient is followed daily, both clinically and by blood smears, to detect the development of malaria infection. 'This is a procedure for testing the ability of native anopheles to acquire and transmit malarias. The results obtained assist in appraising the public health significance of native malarias as well as of foreign malarias which are introduced.
So the answer to the question was yes. Domestic mosquitoes were able to transmit foreign malarias. The returning servicemen were not going to be a dead end for these introduced parasites. These are just numbers from one paper for one parasite. They're just relative numbers of the ability of these various mosquitoes to transmit. So the mosquito at the top is 50 times better than the mosquito at the bottom. But what I think you really need to just take away from this is that there were plenty of domestic mosquitoes that were able to transmit these malarias, and that in particular the first and third are our two major domestic vectors. So this was a real public health threat. What was concluded from this? Well, from the positive result meant that they had to continue the public health measures they had underway already to deal with returning diseases. And that was primarily twofold. One was the random testing of servicemen on troop ships on their way back to the States to make sure there weren't outbreaks that they weren't finding. And secondly, a public health education effort among the servicemen themselves, so they would be able to recognize the symptoms of disease, let them know about the possibility of relapse, and secondly, an information program for U.S. physicians, particularly those who practiced in areas without endemic malaria, so that they would be able to recognize this disease if it turned up in their practices. Now I want to move forward in time, almost 20 years. We're going to focus on the Kuala Lumpur field project, but before we go there, we have to stop on our way in Memphis, Tennessee, to see if we do have a new public health emergency emerging. It's the spring of 1960. Don Iles, who I introduced to you, I'm going to show you two quotes here from his wife's memoir. Mary Iles wrote a memoir. He was sick, and he had fever and chills. He had been to the doctor. They weren't sure what was wrong with him. His wife thought he had malaria. He, although he was a malariologist, didn't think he had malaria because he was living in Memphis, Tennessee, where there was no endemic malaria in 1960, and he was working with monkey malarias, which were not transmissible to humans. But he was a good husband, and when his wife told him to get a smear, he did a smear. She was right. He did, in fact, have malaria, and he had become infected with his monkey malaria that he was working with. Granted, they were somewhat cavalier in their handling of these mosquitoes, but they were working with thousands of infected mosquitoes, but they didn't believe this was a transmissible disease. So the first thing they did once they had determined that Don had monkey malaria was to infect two more members of the laboratory staff to determine the typical self-experimentation that this wasn't just a fluke. Then they sent the parasites out to Bob Coatney, who was Don Iles' boss here in Bethesda. He took them down to his research program in Atlanta, where he worked with prisoner volunteers. This is a picture from a slightly earlier period, and unfortunately Bob Coatney is the guy in the back that you can't quite see. But he jumped on this, as Mary Iles suggested. He confirmed their findings that indeed mosquitoes could transmit this parasite, this monkey parasite, to humans. And as she suggested, he published this. So this was the monkey parasite, Plasmodium cynomolgi bastianelli. This is an illustration from a lovely book on the primate malarias that they published some years later. And then the question for us today as historians looking back, well, we wonder, well, was this, bless you, was this an isolated lab problem, something to be corrected by just improving their handling procedures, or was this a serious threat to public health? Well, because it was 1960, you were 5 years into the World Health Organization's malaria eradication program. And if you want to eradicate a disease, you can't tolerate, you can't do it if you have a reservoir of the disease in animals. So, for example, in South America and Southeast Asia, there are large populations of monkeys, all of whom have various monkey parasites. If these can jump from monkeys to people, eradication is in deep trouble. So the basic question then came to be, could that happen? Was that happening in the natural environment? Was malaria a zoonosis? This is a quote that I'm actually going to read to you. This is a quote from an oral history I did earlier this year with Mac Warren, who was one of the scientists who was sent to Kuala Lumpur to look into this. Now we suddenly realize that here's some place in Southeast Asia with huge populations of monkeys and at least one monkey malaria parasite that would infect man. How many of these were there? How many parasites were there out there? What was going on? What was this going to do to the concept of malaria eradication? Geneva, where the World Health Organization is based, London and Washington were really very spooked about the whole business, because suddenly, if there was a significant level of malaria in non-human primates infectious to man, then the concept of eradication had biologically gone down the tubes. There was simply no possibility that it could be eradicated. Bob Coatney jumped on this. He had a lot of expertise, and his people did, in primate malaria. They got right on this. As Mac Warren said, the NIH was prepared to invest funds into sending this team to Kuala Lumpur to begin to investigate monkey malaria. What was it doing? How much of it was there? What were the possibilities of having it be transmitted to man at any significant level? So what I want to do is just quickly take you through the why, the where, the who, the research possibilities, and the actual, a brief sketch of the kinds of research they did conduct. The why I think we've covered. The threat to malaria eradication was the why. The where was Kuala Lumpur, Malaya, and I'll probably slip back and forth between saying Malaya and Malaysia. During this period, Malaya was undergoing the process of becoming an independent country and no longer part of the British Empire, and the name changed, but it's still the same place. There were a number of reasons why Kuala Lumpur was a good site. First and foremost was the presence there of the Institute for Medical Research. This was a leading center for tropical disease study. The World Health Organization, the U.S. Army, other international research programs had programs at the Institute for Medical Research, and there were also local experts. Kuala Lumpur was a good choice because the monkey parasite that Don Iles had become infected with, Plasmodium cynomolgi bastianelli, was native to the Malay Peninsula, so they knew they had a potential locus right in the neighborhood. And also, remember, this is the early 1960s in Southeast Asia. Malaya was a relatively stable country politically at the time, so it was a relatively safe place to send people to. And finally, Malaya was not participating in the malaria eradication program, which meant that people and resources would be available to support this program, and not all those resources would be tied up with the eradication program. There were a whole series of these pictures taken. As I say, it was a very active research center. People would come through to lecture there, and when they did, the director of the institute, Dr. Sandosham, would get everyone out in front for a photograph. And just two things I think I'd like you to take away from the photograph. One is a fine colonial imperial institution in a time of transition, and also just the diversity of the people who had come to work in this place, that it was a very exciting and lively research environment. The WHO I've already mentioned. Don Iles, who was infected, also early on led the team in Kuala Lumpur. Mac Warren joined the Public Health Service in order to work on this project, and at any given time they had 1 or 2 US technicians rotating through the lab there. They also had local people who helped them, and other foreigners, and one in particular I guess I should mention is Harry Wharton, their Australian entomologist, who was very central to this work. This is a picture of Don Iles with his customary cigarette. I show this picture because I think it encapsulates something about the man. He was not just a malariologist of 2 decades experience. He was also a botanist, a naturalist, and interested in all sorts of natural history and ecological pursuits. So he was exactly the kind of person you'd want to send out into the field to look into such an open, essentially ecological question. I'm not sure what he was collecting on that day with his gun bearer. This is what Mac Warren had to say about Don Iles. He was arguably the most accomplished, the most brilliant scientist thinker that I ever had the opportunity to be around. We were committed to finding out about monkey malaria, and nothing was known about the epidemiology of monkey malaria, so the world was our oyster. There's a tragic note to this story. Don Iles, as he was leaving, literally as he was boarding the ship to leave Malaya to come back to the States, died of a heart attack on the boarding ramp and left his wife, Mary, and 3 children behind. But that tragic note aside, I wanted to just focus on the last sentence here. So the world was our oyster, because I think it points to the research possibilities that were in front of them when they arrived in Kuala Lumpur. As Mac Warren said, there was nothing that we could do in the jungles of Southeast Asia that would not be pertinent to finding out about monkey malaria. And that's primarily because so little was known. But what I wanted to take you through is what they did do, not all the possibilities that they didn't pursue. And I wanted to start with the monkeys and peoples question. At heart, what they were trying to determine is if monkey malaria in a natural setting was being transmitted to human beings. That's a fairly complicated question because for that to happen, you have to have infected monkeys, you have to have people who sleep exposed or somehow exposed to the mosquitoes who are nearby, and you need a mosquito who will commonly and by choice feed on both monkeys and people, and also provide a suitable host for the parasite. So it's a fairly complicated ecological and geographical question. So they needed some resources to look into this. In the laboratory, they needed uninfected monkeys to experiment with, clean monkeys. Again, that's a geographic question. This is not a historically accurate map, but in the middle left, you can see the Malay Peninsula. You can see Kuala Lumpur, nicely located for access to much of Southeast Asia. But on the questions of clean monkeys, that's a geographical question. You have to get beyond the range of these monkey parasites, and that means you need to get your monkeys either in northern Thailand or pretty high up on the Indian subcontinent. So where they went is just off the upper left corner of this map. You can just see protruding into India there, what we today call Bangladesh, which was then East Pakistan. And they would go there to get their clean monkeys. This is a picture of Saeed Mujibullah and Mac Warren. Mujibullah was their monkey dealer there, but he was more than that. He was in a way a collaborator. He had his own microscope. He was interested in the project. And he also helped Dr. Warren out when he came down with malaria on a visit to East Pakistan. Beyond the monkeys, to do primate research, they needed insectary facilities to raise the mosquitoes, much along the lines that I already outlined for the Columbia project. And then there was a large field component to the research, and I'm just going to give you a flavor for that. You need, as I suggested, to collect mosquitoes, but also to observe their behavior, to figure out where they breed and where they feed, and were they potential vectors to carry a parasite between monkeys and people. So to catch mosquitoes, you need to be able to trap them. They tried a number of different traps. You can pretty much make that out. You put a small animal where it says bait, and then as mosquitoes were attracted to this animal, they'd be sucked up by the fan through the funnel and into the collecting bottle. This system they tried, and they didn't like it. It didn't work very well in Malaya. And also you need something to drive the motor. A much simpler mosquito trap is the dry ice lard can mosquito trap. You put dry ice in it, which generates CO2 to attract the mosquitoes. They come in through one of these funnels, which you can cut to suit your mosquito variety. They come in through the funnel and can't get out. It's a typical flying arthropod style of trap. And finally, when neither of those kinds of traps worked particularly well in their environment where they were conducting this research, they went to the net-baited traps. And this is primarily what they used to collect their mosquitoes. It worked best. They had a seamstress to sew up these traps. And if you just look inside there, you can just make out, I think, 2 monkeys in the box. They used 2 or 4 monkey baits. And if you were experimenting to see whether the mosquitoes were coming to feed on humans, then you could sit in the trap and be the bait. But basically, these were set up to collect mosquitoes at night. That's primarily when they feed. And the way it would work is you'd set this up with a couple of monkeys in it, and someone would come every hour with a flashlight and an aspirator bottle and collect the mosquitoes from the inside of this net who had come to feed on the monkeys. And of course, monkeys don't generally sleep on the ground. They sleep in the trees. So you also had to set up these traps in platforms. And I gather it was sometimes a harrowing experience to be up on that platform in the dark with your aspirator bottle and your flashlight to collect the mosquitoes. There were also other hazards. Well, mosquitoes aren't the only thing that feed on monkeys. Sometimes they'd hear the monkeys screaming at night and a python had found its way up there and was trying to get into the trap. Or on your way up or down this ladder with the aspirator bottle and the flashlight, you'd encounter the python that was on his way up or down. So it was an interesting kind of research, very different from what you might encounter in the laboratory. Now, as I say, they're interested in locating where the mosquitoes breed and where they feed. In one of their research areas, the breeding locations was in the Nipa palm groves. These are plantations. And at the base of the palms, you can just about make out these structures. They catch water and they provide a breeding place for this particular species of mosquito. This is sort of a back shot of the Nipa palm plantations. The palms are used for making thatch for roofs. So here we have a map of a research area. I'm just going to, as I say, try to sketch out what they did here. There's something I think particularly lovely about this piece of research. Basically in area A, you have, if you can read you have Nipa palms growing there. And so this is where they're breeding, but they're not feeding there. They find the mosquitoes feeding largely out in the mangrove swamps that surround this. And then they had to answer the question, well, are these really the same mosquitoes, or are we just missing where the feeding mosquitoes are breeding? And the way they answered this question is you can label your mosquitoes using finely divided metal dust. So gold dust, silver dust, bronzing powder give you 3 different colors for labeling your mosquitoes. And they took either mosquitoes that they had caught in area A or their own freshly raised, unfed mosquitoes from the laboratory, labeled them in this way, released them in area A, and then had a set of traps extending out to try to catch them. And what I think is beautiful about this experiment is they released 205 mosquitoes and actually recovered 9 of them, which given the size of the mosquitoes and the size of the research area is impressive. And even more impressive when you consider that some of the mosquitoes were recovered more than 500 yards away out in the mangrove. So they disperse over a very large area. And obviously that sort of thing, like how far a mosquito will fly to feed, is important if you're trying to figure out, is this a potential vector? Of course, you want to know what kind of parasites these mosquitoes might be vectoring for you. So they have to collect monkey parasites. This was largely done by buying monkeys from local people or from dealers and then checking their blood for parasites. They discovered 4 or 5 new monkey parasites, including this one, which they named in memory of Don Isles, Plasmodium ilesi, which they took out of a white-handed gibbon. Finally, they wanted to look in human populations to see if there were monkey parasites in these populations. They wanted to look in populations where people were sleeping in the open, where they were exposed to mosquitoes, where there were monkey populations nearby. So it took them to some fairly rugged terrain, and also to some places, you know, in the early 1960s, like the border between Vietnam and Cambodia, which were politically sort of interesting at the time, among the Montagnards, who you may recall were U.S. allies during the Vietnam War, and across a number of countries in Southeast Asia looking. And these, when they collected blood there, it was more than just the diagnostic finger prick to determine if people had parasites in their blood. They actually took aliquots of blood by venipuncture and then pooled 5 of these samples at a time and injected those pooled samples into the monkeys. And they often collected from hundreds or thousands of people and then injected these into clean monkeys, these combined aliquots. And what they found was nobody infected a monkey. None of these people had monkey parasites in their blood. And so the conclusion was that although the simian malarias of Asia might occasionally infect somebody, and there are a few documented cases, they were not a serious public health problem. So then what did this mean for eradication? Well, by the end of the project in 1964-65, it really meant very little for eradication because eradication was already on the ropes because of 2 other problems, that is DDT resistance in mosquitoes and drug resistance, especially chloroquine resistance, in the parasites. So here you have a negative finding, but by the time it's reached, it's essentially besides the point. So what do I think these 2 stories tell us more generally? I think they point to how research questions become urgent or important or interesting or hot or whatever the terminology you want to use. And they also tell us about the converse, how a research question for exogenous reasons can suddenly become unimportant. I think they tell us something about the complex relationship between biomedical research and public health and how these 2 areas interlace and drive one another. And I think they also yield a novel view of the relationship between center and periphery, between developing countries and developed countries, about where resources are, where expertise is, and where problems and their solutions come from. And I'd like us to take 2 historical lessons away from this. First, as I suggested at the outcome, that these are part of a deep culture, a long history of public service and a flexible response to health emergencies that are really part of the culture here at NIAID and NIH. And secondly, if you remember what I just said a little bit, you can think back. I told the stories in 2 sort of different ways, 2 different voices, if you will, and partly that was driven by what sources were available, what had survived from in the records of these 2 programs. And I think people at NIH are making history every day, and we all need to think about what papers, what artifacts, what objects pass through our hands every day that are part of that history and need to be preserved. If you want to know more about that, we have all kinds of information at the History Office. But I think the ability to be able to recapture this history is very important, and we need to think about it today. I just want to close by thanking NIAID and Dr. Zun for supporting my research. I want to especially thank Dr. Miller, who pointed me towards Martin Young and Don Iles. He pointed me towards several other people on here, Bill Collins, Jeff Jeffrey, and Mac Warren. I did oral histories with the 3 of them, and that really gave me some context and background and some of the personal experience that informed my understanding of these research projects. And Bill Collins is a great collector. He supplied me with the movie and with many of the pictures I had today. I want to thank Dr. Harden, the NIH historian, for supporting me and housing me in the NIH History Office. And I want to thank my NIAID scientific mentor, Dr. Tom Wellams, for his support and his kindness throughout this project. And thank all of you for listening.
Now I'm on. Okay, thank you so much, Leo, for a fine talk, and I want to thank the audience for coming. I hope you all will come and join us for a small reception out in the lobby, courtesy of the Foundation for Advanced Education in the Sciences. And you can ask Dr. Slater some more questions or talk to somebody about what you might like to donate also about your research. Thanks again for coming. We are adjourned.